Hi everyone. Our next topic is perception and action. One of the reasons I chose the textbook for this course that I did is that it has a chapter on perception and action. Most textbooks on perception do not include a chapter like that. Largely in this course, we are considering input, taking in information, but there also has to be an output. What's the reason why we're taking in all this information, if not to guide our actions? So we'll explore a little bit about action. Here are the learning objectives. I'm going to be talking quite a bit about J.J. Gibson's ecological approach to perception, because this is an approach that did firmly consider the role of action and behavior. It wasn't just kind of statically trying to describe perception, but tying it instead to the output in the things that we do. So I'm going to be elaborating a little bit on J.J. Gibson's ideas, including the idea of direct perception and invariance. Also, affordances. After explaining a bit about J.J. Gibson's ecological approach, I'm then going to turn from this kind of theoretical approach to a more neurological approach. I'll be taking a look at some areas of the brain, intraparietal areas and their functions. Here's a hint, it's going to involve action. And I'm going to be spending a lot of time on something called mirror neurons. I've also got a video for you to watch on mirror neurons. We'll talk about the functional properties of mirror neurons, and then a bit of extrapolation, perhaps. I'll talk about how V.S. Ramachandran uses mirror neurons to account for technological advances. But first, the ecological approach. This was established by J.J. Gibson basically single-handedly. J.J. Gibson described this as sort of a behaviorist approach to understanding perception, which is a weird mashup of those two terms. The behaviorists largely viewed the mind as a black box. Whatever happened inside there doesn't really matter. It just matters what the stimuli are and then what your responses are. So input and output, stimuli and actions. Here are some of the basic assumptions of Gibson's ecological approach. It's an ecological approach. I know that's obvious. What do you think of when you hear the word ecology? Do you think of the environment? Yeah, it's literally the environment, one, that we evolved in. So it's about understanding our perceptual abilities in the context of natural selection. Our natural environment is also important. We have evolved a particular way because of the environment that we evolved in. And lastly, we move through our environment. Motion is very important to perception. At the time, a typical experiment in perception would involve going into a dark laboratory, having your chin in a chin rest. They'd strap your forehead in, they'd tell you not to move, and they would flash an image on a screen for a fraction of a second. Gibson said, that's not the way we see, and you're really kind of losing the fundamental nature of perception. Second basic assumption. The world is rich with information. The information that enters our eyes is incredibly rich. We've got a light source, it's bouncing off of surfaces according to geometry, going into our eyes. Really, the job of vision is to pick up this very rich information. This is very much a bottom-up approach. Gibson's approach is essentially the ultimate bottom-up approach. Gibson referred to things like the optic array. That is, if you imagine light as rays of light being emitted from a light source, bouncing off of a surface, bouncing into your eye, that collection of all of those rays of light is the optic array. It's not hodgepodge, it's not random, it's highly structured and organized. All you have to do is pick up that information. That's all there is to it. I've spent a lot of time talking about edge analysis and detection, object surfaces, all kinds of things. Gibson said, you don't need that. That's not how vision works. It's not about breaking things down into bits and pieces. That's not how we see. You just open your eyes and you see. Now, that being said, there are certain relationships in our environment that are invariant. Now, this makes the job of explaining perception a little bit easier. Our visual system will pick up on invariant properties. Now, be careful, this doesn't mean that what we're looking at is static and unchanging. That's not what it means. It means that there's a relationship among things at a higher level. This relationship is constant. An example of this is the texture gradient. If I show you this image, which I've shown you before, well, on the screen, their shape is changing a little bit from top to bottom, and their spacing is changing. But if you imagine this to be a texture that's consistent, and it's on top of a three-dimensional curved surface, well, then it's completely obvious what it is. The texture gradient means that as things are closer to us, we can make them out in greater detail. They are larger on our retina, and as things fade off into the distance, they get smaller and closer together and all blend together. That's generally true of the world. That is a constant relationship among elements. Now, of course, motion was important to Gibson, so he coined the term optic flow to the pattern of change that occurs across our retinas. Here's one of Gibson's images. Now, these arrows we can think of as vectors. So this is the view of a pilot who is not coming in for a landing. First of all, we can trace all of the arrows back to where they started from, and that'll be somewhere right about here. It's over the horizon. This is the point that the aircraft is heading towards. They're not coming in for a landing. Now, texture elements that are more to the edges are going to be moving very, very fast. But things that are off in the distance, like these mountains and the texture elements on the ground down here, the arrows are shorter, meaning they're not changing as much. Now, the specific objects that are on your retina at a given moment in time do change from one moment to the next, but the pattern of change is constant. Again, here's Gibson's image flying over a runway. We can tell the difference when we're coming in for a landing because that central spot called the focus of expansion is now down there. Now, don't aim directly for the ground. You don't want to crash into it. The fourth and final assumption is that perception is direct and immediate. This is different from every other theoretical approach I'll talk about in this course. Every other approach is indirect, proposing some intermediate processing. Yeah, the light enters our eyes and then the processing begins and it goes to the LGN and more processing and V1 and more processing. Processing. Gibson said, no, if there's all this processing going on, vision would work like this. You open your eyes and then some time passes and, oh, now I'm seeing something. But that's not how vision works. You open your eyes and you see. It's immediate. So Gibson did not feel the need to propose any mental computation or any inferences or anything else going on. No top-down influence. Everything is bottom-up. Now, Gibson did have to argue with basically everybody else in the field of perception because his view was very iconoclastic. What a good theory needs is supporting evidence. Now, although there is supporting evidence, there's not a heck of a lot of it. But this study is often pulled out as supporting evidence for the ecological approach. Lee and Reddish analyzed the diving behavior of a seabird known as a gannet. So what a gannet does is it flies above the surface of the ocean, and then when it spots its prey, a fish, it'll start to dive. And they're very, very good at diving and catching fish. Now, Lee and Reddish relied on a mathematical construct called tau. And this is an image that, okay, I found it on the internet, okay? If you're into the mathematics of it, there you go, there's tau. Tau is a ratio of your target's retinal size at two different times, divided by the rate of change of its retinal size over time. What? That's a lot of different quantities. Okay, put simply, tau is time to impact. It was actually first proposed in, of all things, a science fiction story in which the main character is looking out through a telescope at an asteroid, and this asteroid is coming towards the Earth, and this person has to calculate when is it going to hit the Earth. And it was based on these values. So if you're looking through a telescope at an asteroid that's coming closer to you, it's going to get bigger. Its retinal size is going to change over time, and this can give you time to impact. And when it comes to the seabirds, the gannets, people made all kinds of proposals about how the gannets do what they do. Does it depend on them starting their dive from the same height every time? And the answer is no. Just simple observation can show you that the gannets start their dives from all kinds of different heights. Tau, interestingly, is an invariant property based on optic flow. It takes into consideration things like terminal velocity and the starting altitude and all these other things. Now, the thing about the gannets is that they always retract their wings right before hitting the surface of the water. Now, they want to leave their wings out as long as they can so they can properly aim and catch the fish. But if they leave their wings out when they hit the surface of the water, they're basically going to break them off and die. So they need to tuck in their wings, but at the last possible moment. How do they do that? Well, they could only do that if they're somehow picking up time to impact. Again, lots of proposals including, well, the gannets always dive from exactly the same height, and it's just a learned behavior, but no, they dive from different heights. They seem to be using tau to resonate to and pick up their time to impact and hitting the surface of the water. Interestingly, some supporting evidence was uncovered in 1992. Wong and Frost found that there was a neuron in a pigeon that fired about 1 second before a simulated collision. They didn't actually throw a ball at a pigeon. That would be mean. No, they set up a screen and they simulated a ball. Basically, it would seem to get larger. So bird would interpret this as, oh, I'm going to get hit by this ball. It's preparing for impact. And they found a neuron in a pigeon that would fire about 1 second before a simulated collision, regardless of the actual size of the object or its velocity. And you can see they manipulated a lot of these. So 10 centimeters at 3 meters per second, it fires about a second before collision. 30 centimeters at 3 meters per second, it fires a burst about a second before. 30 centimeters at 7.5 meters per second, again, it fires about a second before collision. Maybe this neuron is doing tau. Now, any theory of perception is also going to have to explain how we perceive objects, and that was in my last lecture topic. Now, J.J. Gibson had to address this, but once again, he did it in a way very different from every other theory. Now, the other theories that I talked about were all structural in nature. What are the structures? Are they line segments? Are they geons? Are they templates? What are the physical bits and pieces? J.J. Gibson's approach to object perception, in contrast, is functional. Not what it is, what it's made out of, but how does it work? What can you do? And this is where his term affordances comes in. Affordances is the set of actions that you can perform with a given object. So if there's a tree, for example, you can do all kinds of things with a tree, also some things that you can't do with a tree. So it's not about taking an object and putting it into one single category, but coming up with all of the actions you can perform with an object. Everything in our environment has affordances, even the atmosphere. The fact that we have air, it affords visual perception. Visible light can go through the air. Sounds can travel on the air, giving us auditory perception, and even odors can travel on air molecules, so they provide the affordance for olfactory perception. So, trying to come up with the pros and the cons, and I'm trying not to come up with a list of 100 cons, one definite pro is that the concept of invariance has turned out to be very useful to understanding perception, and it has kind of outgrown the ecological approach. It exists outside of this one theoretical paradigm. On the downside, probably the most significant is that perceptual processing does exist. Perception is not direct. It is indirect. We have evidence from all these brain areas that are doing processing. You can't just say, it doesn't exist, not important, don't need to know. No, we actually do need to know how exactly information is being processed, both in a bottom-up fashion and also in a top-down fashion. Now, moving beyond the theoretical approach of J.J. Gibson, I want to turn now to neurons and actions, focusing specifically on intraparietal areas. Now, this brain looks a little bit different. It is a macaque brain. It is not a human brain. So there are some important intraparietal areas that have to do with actions. So I'll go through some of these. There is the lateral intraparietal area. That's the LIP. It's orange, and it's located right there in this brain. This represents the location that you are going to move your eyes to. Did you know that you plan where your eyes are going to move to? Thank your LIP. This is also called the saccade planning area. We have the medial IP, which I can't really show you in this diagram, unfortunately. This represents the area of space that you can reach to. If it's too far away, you can't reach to it, it's not mapped in the medial IP. It's also called the parietal reach region. So again, it's important to know where you can reach to before you're even carrying out that action. Next is the ventral IP, which responds to moving nearby visual input and tactile input to your face. What does that mean? Well, when you're trying to feed yourself, if it's really early in the morning, you got your bowl of cereal and you're feeding your face, you don't have to have your eyes open. You can keep them closed and you can still basically shovel that food into your mouth. That's largely because of your ventral IP. And finally, we have the anterior IP, the AIP. It's kind of a lavender kind of region down here. This represents shape information for representing what kinds of things are graspable. Just because something is close by you doesn't mean you can grasp it. There might be a wall right next to you. You can't really grasp a wall. Interestingly, anterior IP is located between sensory, incoming, and motor, outgoing areas. And in the AIP, there are neurons that respond to both visual stimuli and physical action, tying those two together. Ooh, that sounds pretty interesting. So I want to stick with the AIP a little bit. Now, there have been several different types of AIP neurons identified. One is called motor dominant. Now, these ones fire very strongly when an action is performed. It doesn't matter if the lights are on or off. It doesn't really matter. But the action has to be performed. If you just see a visual cue, that's not enough. It's all about motor actions. Visual dominant, slightly different. They respond when an action is performed in the light, so you can see your action being carried out, but they do not respond if that same action is carried out in the dark. So it's all about vision, being able to see what's happening. We also have visual and motor AIP neurons. Now, they respond most strongly to an action performed in the light, but also to a lesser degree to that same action performed in the dark, or to just the visual cue happening all by itself, seeing an action being performed. We also have some nearby neurons in the intraparietal sulcus or groove that are implicated in some cognitive functions that require some integration of sensory and motor information. So again, putting together some input and output. Now, of course, this is not the end point. From these regions, there are projections that go to the premotor cortex and then to the motor cortex. So again, they're sifting through incoming visual information and tying it eventually to output or motor actions. And it's in this context of the study of these individual neurons and these regions of the brain in macaque monkeys that I want to introduce Giacomo Rizzolatti and his colleagues. For their work through the 1990s, they shared their Groemeyer Award in Psychology in 2007. This is one of the top awards in psychology. Now, what Rizzolatti and his colleagues were doing was probing the premotor cortex. This is called area F5 in macaque monkeys. So again, I have to point out, F5 is not in a human brain. This is in macaque monkeys. Now, they're able to identify a neuron that would fire strongly when the monkeys grasped a piece of food. Here the story turns a little bit. No one can quite remember what exactly the piece of food was, but they weren't doing any active recording. They kind of had taken a break. This is very painstaking work. The monkey was still in the apparatus, and the probe was still in its brain. And one of the researchers reached for some food that was nearby. Rizzolatti remembers it as a banana, but others remember it as a peanut or an ice cream cone. Anyway, when they just reached for this food that was sitting there, in view of the monkey, the neuron fired very, very strongly. Now, the monkey was just passively watching this, but its neuron that previously responded to action now fired very, very strongly. And the term that was coined for this neuron was a mirror neuron. A mirror neuron is a neuron that's activated when an action is performed or when an action is observed. This neuron has one foot in each of two different worlds. I know I'm mixing my metaphors, sorry. There was an explosion of research trying to identify exactly the role of mirror neurons. They found mirror neurons for grasping, manipulating, or even placing objects. So what we see here is the firing rate of some mirror neurons. In this case, the macaque would be watching a human pick up, I don't know, is that a peanut? Something like that. And it fires pretty strongly to that. In the second case, it's the monkey itself picking up the peanut. Fires a burst of activity. But over here, there's a human using pliers to pick up a piece of food, and there's really not much activity. Again, this is one neuron, or a set of neurons, that's firing in these three different response patterns to three different experiences. I have a video for you to watch about the discovery of mirror neurons. Now, since the time of Rizzolatti and his colleagues, again, there's been an explosion of research into mirror neurons. So here are some features of mirror neurons. They require something called biological motion. And in the image that I showed you a couple of slides ago, if you pick up a piece of food with pliers, the mirror neuron doesn't care. It's kind of a mechanical motion. It's not a motion of a biological being. It's not a monkey, it's not a human. What is a mirror neuron? It seems to be matching execution, that is, performing an action, and observation, or perception. They found mirror neurons that have been activated as novice guitarists watch a professional play chords on a guitar. Mirror neurons do not like mime. No, there has to be an object present. So if you mime picking up a non-existent piece of food, you know what? Like many people, mirror neurons don't care. If you just show an object, mirror neurons don't care. They're not about detecting objects. No, they're much more sophisticated than that. There are audio-visual mirror neurons. They're activated by visual information and the sound of the corresponding action, like seeing paper being ripped or hearing the sound of ripping paper. I was going to rip some paper for you, but I don't have any paper to rip. I need all these pieces of paper. It sounds like this. Like that. Okay. Now you have to keep in mind that mirror neurons were conclusively identified in monkeys, area F5. We don't have area F5. But an important question to ask is, do we have something that corresponds to F5? It's still the subject of quite a bit of debate, But some people suggest that Broca's area, which is important in speech production, is the counterpart to area F5. Clearly, monkeys do not speak, they do not understand speech, not like we can, so it's not exactly the same kind of thing. There are some theories about speech perception that say it's important to be able to pick up speech, that is, to perceive speech in order to produce speech, tying together perception and action. It's a very tantalizing conclusion. So a lot of the research has shifted over to human beings looking for mirror neurons in humans. We have found areas of the premotor cortex that get activated when people read a literal phrase, biting the peach or grasping a pen. They will also become activated when you watch a video of a peach being bitten or a pen being grasped. Interesting. So it looks like, again, they have a foot in two different worlds. Interestingly, these areas are not activated by metaphors, like biting off more than you can chew or grasping the idea. They're smart enough not to get fooled by these things, knowing that they're metaphorical and not literal. Mirror neurons may also even infer the goal of an action. Any action must be performed in order to accomplish some kind of goal. Let me explain the research. So, first of all, you put some food behind a screen. The monkey sees this happening. Then a human model reaches for some food, but the end of this is blocked by the screen. Oh, they're going for this piece of food, but you can't actually see the hand grabbing the food. Nonetheless, this is enough to activate the mirror neuron. The mirror neuron knows why the hand is going behind the screen. Mirror neurons may also understand even higher level intentions, which are called hypergoals. So a hypergoal is, for example, picking up a mug because you want to have a sip of the liquid, or picking up a mug because you're cleaning up. So mirror neurons, when presented with videos of these two actions, can differentiate between the two. They know that they're not the same, even though the motion of the hand grasping the mug is identical in both of these. The hypergoal is very, very different. That is some amazingly sophisticated processing on the part of these neurons. Now, I did mention biological motion. I do want to come back to that again. In this study, participants had their brains scanned in an fMRI while they were watching videos by a robot endoskeleton, creepy, a human-looking android, or an actual human. Okay. And it turns out there's a difference in activity. So, yeah, in parts of your brain, it knows the difference between a robot endoskeleton, an android, and a human. Well, the android and human produce pretty similar activities, but the robot, that was clearly different. And I have the actual brain scans from this study to show you. So watch out for those endoskeletons. Never know what they're up to. They're a bit tricky. Now, this study was very careful to say that the activity may be due to the human mirror neuron system. Why didn't they come out and conclude this definitively? Mirror neurons have been identified by single-cell recording. Doing an fMRI is really not the same thing. Moving 1 millimeter or another in the brain can make a huge amount of difference. So that's why these conclusions are a bit tentative. In order to definitively establish mirror neurons, there would have to be intracellular recordings done on human brains, which is pretty tricky and difficult to do. Nonetheless, the evidence does seem to be converging that we probably have these mirror neurons as well. In fact, some people have taken the idea of mirror neurons and have tried to apply them. So what you can do is program them into a computer to simulate their action. This horribly creepy looking thing is called the iCub. It is an open source robot, and it is programmed with visual vestibular or balance, auditory, and haptic sensory capabilities. It has been programmed with the equivalent of mirror neurons. So I'm going to show you a video in a second of the iCub. The iCub is actually mimicking the actions of a person sitting in a chair. It hasn't been pre-programmed with these actions. It's just been told, okay, perform the same actions that you see the person performing in front of you. So check out that video. I don't know why they had to make the iCub look so creepy. Of course, mirror neurons also raise a lot of further questions. So I don't have answers to these, but people are still working on these. Maybe mirror neurons can answer some big picture questions. We can understand the actions of other people, even their intentions. We can learn by imitation. We can share attention. Are mirror neurons the things that underlie all of these processes? Mirror neurons don't exist by themselves. They're not an island. They get connections from other parts of the brain. What other parts of the brain exactly? How is the functioning of mirror neurons modulated? Where and how? We'd expect neuropathological evidence. That is, if a person's mirror neuron system is damaged, then they should have difficulty understanding the actions of other people. Even bigger picture questions include, does the mirror neuron system underlie human language, something that we learn effortlessly as children? Is it because we can learn by imitation? We can watch other people speaking and then just pick it up automatically? What about theory of mind? Okay, that's not the theory of the mind. That kind of sounds like what psychology is all about. No, this is a different concept. I need to go into a bit of a detour on theory of mind. Theory of mind means that you understand that other people have minds of their own. Other people have their own beliefs, desires, and intentions. Can you put yourself into the shoes of another person? You can test theory of mind through something called the Sally-Ann test, which is a kind of a little skit that is shown to the people who you are testing. So it goes something like this. Okay, you have a couple of dolls. One is Sally and one is Ann. So Sally has a basket. Ann has a box. Sally puts her ball into her basket and then she leaves. But Ann is still there with the box and the basket. Ann, being a bit mischievous, takes the ball out of the basket and puts it into her box. Now again, the people that you're testing are watching this happen with these puppets and everything. The question that is posed to the participants when Sally returns is, where will Sally look for her ball? It's not asking, where is the ball? So there's two different answers. Of course, the ball is in the box. Where will Sally look for her ball? The correct answer to that would be, well, she should look in the basket where she left it. But can you put yourself in the shoes of Sally? Okay, here are some typical results comparing neuronormal with Down syndrome and people with autism. So this is reported in terms of years, months. Sometimes in doing research with children, a couple of months can make a big difference, so that's why it's reported in years and months. So for a neurotypical child of about 4.5 years of age, they get about 85% correct, so pretty good. For children with Down syndrome, they have to be almost 11 years old before they get a comparable percent correct. And here, children with autism, almost 12 years of age, they're only at 20%. It seems they have difficulty putting themselves in the shoes or the mind of another person. So here's a study by Jacoboni from 2005, an fMRI study of children, and these faces should look familiar to you if you watched that video. Autistic children in this study had lower activity in regions where we would expect mirror neurons to be, in the frontal lobe, compared to neurotypical normals. Among normals, the amount of mirror neuron activity was correlated with scores on a test of empathy. Of course, we have to be careful with correlations, but this is a very tantalizing finding. This has led some people to call mirror neurons the Gandhi neurons because they seem to be involved with feeling empathy, not just sympathy, but being able to put yourself in the position of another person and feeling what they feel. Now, the person who came up with this Gandhi neuron concept is actually Villanur Subramanian Ramachandran. In 2000, he discussed mirror neurons in an invited talk. This isn't peer-reviewed, so it's not published in a scientific journal, but he was willing to put his name on this, so I'm going to talk about that. Now, Ramachandran is a medical doctor as well as a neuropsychologist studying people who have brain damage. So, if you have a stroke in your right hemisphere, it's pretty common to have paralysis somewhere on the left side of your body, maybe your left arm or the left side of your face. Interestingly, Ramachandran came across some patients, about 5% of people, who deny their paralysis, called anosognosia, and deny paralysis of other patients too. So they have had a stroke, they can't move their left arm. He sits down beside them and says, "Good morning, could you raise your left arm for me?" And the arm just sits there, it doesn't move at all. And they go, "There, it moved." And they'll also claim that other people, they often are in the hospital, and they're with people who have also suffered strokes, and they'll say that other people are moving their arms too, but that's impossible. Ramachandran started to wonder if what was partly damaged in these people was their mirror neuron system. Ramachandran has speculated about evolution and the brain and the role of mirror neurons. So here's some interesting facts. The hominid brain reached its present size 250,000 years ago. Okay, not exactly recently. So in other words, human beings have had a lot of potential for 250,000 years. But many things that we attribute as definitively human, tool use, creating fire and art and music and language, didn't emerge in lockstep with the brain increasing in size. It took much later before these human attributes emerged. Why is that? If our brain seemingly had the potential by virtue of its size 250,000 years ago. We can look to the record of tools created by people. So here's some pretty crude tools from 2.4 million years ago, discovered in the Olduvai Gorge. I know if I was out walking and I tripped over one of these, I'd say, "Look, a rock." But apparently this is a tool made by our ancestors. Now, it is pretty cruel, and we can see upgrades to these rock tools. This is a symmetrical tool from 1.4 million years ago, much better for cutting. And how about this? Bifacial tools with a handle attached, those emerged only 200,000 years ago. And when they emerged suddenly. And I know it's weird to think of rocks as technology, but these are a form of early technology. And when one new type emerged, it spread instantly. Why did they appear so instantly and just take over the previously existing technology? How did that happen? There's something called the Big Bang of evolution, or the Great Leap, when we see cave art and clothes and dwellings, and this happened very, very suddenly about 40,000 years ago. Why did that happen so suddenly that it's called a Big Bang? What about the nature of language? Did language appear suddenly out of nowhere? Did it come from grunts, or did it evolve from a gestural language? Why are human beings called a Machiavellian primate? That is, we can read minds of other people to some extent. We know what people are up to. So, for example, if I'm eating a chocolate bar and my kids are around, I'm not just going to leave it sitting out. No, I'm going to take that with me because I know what they might do to that chocolate bar when I'm gone. Now, all of these really fundamentally important changes did not occur in correlation with an increase in brain size or some other kind of evolutionary change. What about mirror neurons, though? Mirror neurons can potentially explain a very rapid spread of technology. For example, you see one, you do one, you teach one. So you develop this incredibly sophisticated tool. Someone else sees it. Oh, I see what you did there. I can make that. And then someone can see that person make that. And the ideas can spread in a viral fashion. Maybe the mirror neurons were there all along. Maybe they were there 250,000 years ago. They just had to be turned on. A switch had to be flipped. Maybe there was an increase in learnability, whatever that means. Mirror neurons are necessary for these advances, but they're not sufficient. It also requires people coming together and cooperating. We need to have a culture, a society, for example, as well. Ramachandran has said, quote, "Mirror neurons will do for psychology what DNA did for biology." That is quite a statement. In 2008, Ramachandran changed his tune on this a little bit. He has now said, quote, "It remains to be seen whether they will turn out to be anything as important as that. But as Sherlock Holmes said to Watson, the game is afoot." Have a look at the E-Class lecture quiz on this topic.